Researchers have reconstructed the brain of Littlefoot, a controversial hominin which lived 3.67 million years ago. Amid a swirling debate over whether the elderly female skeleton is a new separate species, researchers managed to reconstruct the brain. It reveals a small brain combining ape-like and human-like features. Our ability to reconstruct features of early hominin brains has been limited by the very fragmentary nature of the fossil record, said WITS researcher, Dr. Amélie Baudet, who led the research. The Littlefoot endocast is exceptionally well preserved and relatively complete, allowing us to explore our own origins better than ever before. MicroCT scans of the Australopithecus fossil known as Littlefoot shows that the brain of this ancient human relative was small, and shows features that are similar to our own brain and others that are closer to our ancestor shared with living chimpanzees. The team used the scans to reveal impressions left on the skull by the brain and the vessels that feed it, along with the shape of the brain. While the brain features structures similar to modern humans, such as an asymmetrical structure and pattern of middle meningeal vessels, some of its critical areas, such as an expanded visual cortex and reduced parietal association cortex, points to a condition that is distinct from us. The endocast showed that Littlefoot's brain was asymmetrical, with a distinct left occipital ptalia. Brain asymmetry is essential for lateralization of brain function. Asymmetry occurs in humans and living apes, as well as in other younger hominin endocasts. Littlefoot now shows us that this brain asymmetry was present at a very early date, from 3.67 million years ago, and supports suggestions that it was probably present in the last common ancestor of hominins and other great apes. Other brain structures, such as an expanded visual cortex, suggests that the brain of Littlefoot probably had some features that are closer to the ancestor we share with living chimpanzees. In human evolution, when know that a reduced visual cortex, as we can see in our own brain, is related to a more expanded parietal cortex, which is a critical cerebral area responsible for several aspects of sensory processing and sensory motor integration, says Baudet. On the contrary, Littlefoot has a large visual cortex, which is more similar to chimpanzees than to humans. The study also has shown that the vascular system in Australopithecus was more complex than previously thought, which raises new questions on the metabolism of the brain at this time. This would mean that even if Littlefoot's brain was different from us, the vascular system that allows for blood flow, which brings oxygen, and may control temperature in the brain, both essential aspects for evolving a large and complex brain, were possibly already present at that time, says Baudet. Given its geological age of over 3 million years, Littlefoot's brain suggests that younger hominins evolved greater complexity in certain brain structures over time, perhaps in response to increasing environmental pressures experienced after 2.6 million years ago with continuing reduction in closed habitats. Such environmental changes could also potentially have encouraged more complex social interaction, which is driven by structures in the brain, says Baudet.